we're very proud to continue our sponsorship of this great event. And now I'm pleased to present Equalize 2023's keynote speaker, Dr. Sangeeta Bhatia. Dr. Bhatia earned her BS at Brown University, followed by an MS in mechanical engineering at MIT, a PhD in biomedical engineering at the Harvard-MIT Division of Health Sciences and Technology, and an MD at Harvard Medical School. Prior to her appointment at MIT, Dr. Bhatia held a tenured position at UCSD. She and her trainees have launched multiple biotechnology companies to improve human health. As a prolific inventor and passionate advocate for diversity in science and engineering, Dr. Bhatia has received many honors, including the Lemelson MIT Prize, known as the Oscar for Inventors, and the Heinz Medal for groundbreaking inventions and advocacy for women in STEM fields. She is a Howard Hughes Medical Institute investigator, director of the Marble Center for Cancer Nanomedicine at the Koch Institute for Integrative Cancer Research at MIT, and an elected member of the National Academy of Sciences, the National Academy of Engineering, the National Academy of Medicine, the American Academy of Arts and Science, the National Academy of Inventors, and Brown University's Board of Fellows. Dr. Bhatia is a board member of Brown University, Vertex Pharmaceuticals, and the Association for Women in Science. She has an advisory role with Chan Zuckerberg BioHub, Global Oncology Inc., Glimpse Bio, Send Therapeutics, and Satellite Bi. She has a consulting role at Moderna Therapeutics and receives sponsored research funding from Johnson & Johnson. Other than that, though, she's not really involved with a whole lot. So welcome, Sangeeta, with a very impressive resume. We'll pass the mic to you.
Thank you so much. It's such an honor to be here today. I'm really excited to see this group of people gathered together. I'm going to just share my screen. Hopefully this is working. I wanted to just start out by thanking Nicole and her whole team. They've really been trailblazers in this area. And I know that when I was coming up in entrepreneurship, I was always looking for role models in the ecosystem. And so this is such a great way to simultaneously create role models from each other in a cohort of folks that are supporting each other throughout the ecosystem. So I'll just start by saying, you heard my bio, but that's my professional bio. Who I am as a person is I'm the daughter of immigrants. I was born in Boston. I went to high school in the public schools, and I'm a married mother of two teenagers. I started my first company when our second daughter was less than a year old. So hopefully that's just a little vignette that can give you all a sense that there's no time that is not compatible with entrepreneurship. We're just all doing it differently than we're used to seeing. So I'm going to tell you some stories about companies that I've started and some of the lessons that I'm learning as I go. These are some of my disclosures. You heard about them. Please take everything I say related to these companies with the appropriate grain of salt. Okay. So just to start you off and get you kind of in the headspace of the kind of work that we do in my academic lab at MIT that has led to a number of startups, I work on nanotechnology. And nanotechnology as a field really emerged, as many of you probably know, from microchip fabrication. So this is a picture of a microchip inside all of your smartphones. We can now fit 12 billion of these on a footprint of what used to just be one. And the feature sizes on this chip that we can now pattern with light, that's called photolithography, are 5 nanometers and smaller. So we've gotten really, really good at making teeny tiny materials as a field. And that's really been spurred by an interest in speeding computation, which of course it has. What I've been interested in my group at MIT and the center that I run is how to borrow these fabrication tools instead for medical invention. And that's what I'll tell you about today. Just to let you know that this is probably on your radar, in case you are hearing it for the first time, this idea that nanotechnology can enable medical inventions actually came very much to the fore during COVID. There's 3 examples I'd like to share. So the first is that the variant sequencing, when you hear about Omicron variants of COVID circulating, those were sequenced actually in nanopore sequencers. So those are nanotechnology hardware chips. the lateral flow assays, the paper diagnostics that we all got so good at taking, the color on that line is actually structural color from nanoparticles. And of course, many of us received vaccines that were the RNA vaccines that were RNA packaged inside lipid nanomaterials. So these are just some examples of how nanotechnology was sort of ready after decades of investment to converge with medicine and kind of meet a grand challenge in 2019. And the problems that I think about in my lab are similarly grand challenges. I think one of them is liver disease and the other one is cancer. So what I'm showing you on this slide is kind of how we think about all of the tools that have emerged from microchip fabrication. They're at the top of the slide. There's lots of them that allow us to manipulate teeny tiny things. And a picture of the liver, which is my favorite organ. And by using these tools to study the liver, really doing very basic research over the last 20 years or so, we started 3 companies. And I'm going to tell you the story of a couple of them. I think what's kind of been interesting for me as an academic entrepreneur is that these companies brought us into very different spaces and very different business models. And we had to just sort of learn as we go. So one of them, Heprogen, was a tool company. The second was a nanoparticle company, was a diagnostic company. And the third, which I'll tell you about, is a cell therapy company, a regenerative medicine company, so a therapeutic company. And, you know, as an entrepreneur, each one of these has different investors, different business thesis, different business model, different exits for people involved. And so it's on the one hand, really exciting to be in a fundamental area. On the other hand, like you just have to figure it out as you go. And that's, I think, part of being an entrepreneur. What I won't tell you too much about is another set of technologies that have emerged from our lab, but I'd just like you to know about them because we've used these same approaches in cancer. And this has led to nanoparticle inventions that deliver RNA. That led to a company called Impilo, a targeting company that delivers biologics to the extracellular matrix called Matricome Bio, and then a switchable cell and gene therapy where we use heat to turn on and off biologics in cell therapy called Port Therapeutics. And, you know, as I'll show you today, I think what's exciting about being an entrepreneur is once you do this a couple of times, you really start to recognize when something in the lab wants to grow up and be out in the ecosystem and it needs a team and financing at scale. And I think we become more efficient as entrepreneurs after having done this a couple of times. And I'll actually show you this on a timeline. So this is just the first vignette. This is the tool of photolithography applied to patterning cells. So we're shining light on a surface here and making it sticky for cells, for liver cells. And this is a pretty art project. We did it with an artist just to show you kind of how arbitrary this tool can be. And what we used it for in the lab, it became this startup called Heprogen. What we used it for was patterning liver cells in a dish. And it was to solve the problem of predictive toxicity testing. So you have liver cells, you want to test if a drug is going to be safe in humans. These are human liver cells. And the first thing you need to do predictive toxicity testing is make sure those liver cells are happy. And so what I had discovered as a PhD student in rat cells, and then what my PhD student Salman Khatani discovered in his PhD in human cells, was that if we patterned the cells in a particular way and we surrounded them with neighbors, we called that micropattern co-culture, that we could get them to behave in a dish for about a month like human liver cells. And the idea for the company was we could use this for drug testing, for predictive toxicology. And so we set out to start that company, and there were a lot of really interesting things about this journey. I co-founded it with my postdoc, Salman, and our founding CEO, Bonnie Fendrock. We got money from everywhere, accelerator grants. We partnered with business school students to do the early market research, and then eventually became venture backed. And you know, the most interesting thing I think about this journey was that the company didn't end up doing what we thought it was going to do. I mean, they now sell these for predictive toxicity testing, but actually what people use them more for is drug metabolism. So when you make a drug, it gets metabolized in the liver. And I think it's a classic example of, you start your entrepreneurial journey thinking that what the customer's needs are, and actually you learn as you go what they really are, and what your business model needs to be. For those of you who have questions later about this, the kinds of things that we struggled with in the beginning were, is this a service? Is this a tool? Is this a company that discovers new drugs and then has a royalty stream? Like, what is the business thesis of the company? As an academic, was really exciting.
So pretty. Those look orange.
Yeah, but they're not those big.
Can we ask people to mute, please, if they're not other than Sangeeta? Thank you.
Sure. So what was really exciting as an academic is that, of course, when we took this out into the industrial space, this was automated and scaled and miniaturized. And we made some decisions as a company about what the company would do, which was serve the pharmaceutical industry. Then in the lab, you have to decide what you're going to do that's different from the company. And so we drew a very clean line. And in my lab, what we went on to do was do infectious disease modeling of these. So we studied human liver infections like hepatitis B and hepatitis C and liver stage malaria. And it became a wonderful synergy between the company now making these at scale and being a product that you could buy, and then us continuing to do the sort of fundamental work that we could publish in the literature. When people got excited about the tool, we would say, like, go buy them from the company. So that was a really kind of synergistic and not competing outcome. But it is important to sort of navigate that carefully as you think about launching a company from your lab. The second example I want to give you is more at the nanoscale. These are nanomaterials that we invented that you're seeing here in this graphic travel through the bloodstream to make a diagnostic measurement inside the liver. So the way these materials are designed is when they get inside the liver, they are activated by disease-associated enzymes. And you see these enzymes here in yellow. And they're designed to encounter these sensors and give off a flurry of chemical reactions and produce a tiny little reporter that is not degradable in the blood. You see it here being concentrated by the kidney into the urine. And so the way these materials work is you give an injection of the larger nanomaterial. It makes a measurement deep inside a tissue like the liver that you would normally have to biopsy. And an hour later, you get a reporter in the urine. So you get a non-invasive urine test of what was going on deep inside that tissue. Hopefully that video came through on the, yes, I'm seeing some notes, great. So this is, again, an invention that happened in the lab. We actually invented this first for cancer. And as I was saying before with the other project, what we realized was that this could be really powerful for liver disease. And that kind of aha moment came to me one day when I was hanging out in liver clinic on sabbatical with some colleagues, because I just wanted to see what had happened, to the liver field since I had graduated med school. And every single patient that they saw us, they said, we need to do a liver biopsy, come back in a year. And people said, I don't really want a liver biopsy. We thought, like, oh, this would be a great way to see inside the liver without a biopsy. So we spun out a company to take that forward to patients. And I spun that out with my then postdoc, Gabe Kwong. And this company was a little different because we had no founding CEO like we had in the last one. And Gabe, unlike Salman, wanted to be an academic. So he was like, I'll help you get the company started, but I'm on the faculty market. And within a year, he was gone. He went to Georgia Tech. He's now tenured. He's doing great. But the founding team was totally different. And so what I ended up having to do for this company was actually take a sabbatical and get the company going, raise the venture round myself, put the board together, rent the space in the incubator, all of that stuff. And again, trial by fire. So this company is called Glimpse Bio. They make these ultra-sensitive nanosensors, actually panels of them, for different diagnostic applications, and then they use machine learning to make the diagnosis. And again, the idea for the company was to replace a biopsy with a non-invasive tool. This one, I actually think this company was like my third baby. I mean, this company kept me up at night in just the same way a newborn does. And I really wanted to get it going to the point where, it could walk and talk on its own. So we raised the Series A in 2018, raised the seed in the Series A myself. 2018 was my sabbatical year, and hired a CEO. And then they stepped into the company and grew it and did the Series B, and it's been on its way, completed the first phase 1 clinical trial, and the first clinical data is now coming out. So here again, we took money from anybody who would listen, MIT, angels, venture, grants, pharma, all the things, to get going. And as I said, we have pivoted a bunch of times, and I think that's a really important part of learning as you go. And the last thing I'll say about this company was this one also had a what is the business model question, because if you really think about it, it's an injectable, which the FDA might look at as a drug, but it's a diagnostic readout, and diagnostics are actually regulated in CDER, which is the device division. So what is it? Is it a drug or a device? And people said to us in the beginning, like, you've invented the worst of both worlds. You have drug development costs and diagnostic upside, congratulations. And so one of the things that we had to do when I was there running the company was actually kind of hack that, and convince people that we could get regulatory clarity and we could be actually regulated by the device division. I actually went to the FDA myself and took that meeting, and came out with what's called a request for designation. So we had clarity for investors about what the path was going to be. So that's example number 2. I'm only going to do 3 examples. So the last example is on the cell therapy side. So this is basically using all those same lithography tools that I showed you in 2D and 3D, where we can now photo pattern or micromold ensembles of cells, these living hepatocytes that you see in these pictures in green, and try and use them as therapeutics for patients who can't get liver transplants. And so myself and a colleague, Chris Chen, had been working on this in our labs for about 20 years. And right before COVID, one thing that happened in the sort of external ecosystem that some of you might know is that CAR T therapies got approved. And when cell therapies got approved and reimbursed, we thought, like, okay, maybe the market is now ready for this idea. So we spun out the company together with our trainee, Arnav Chabra. And a common theme that you'll see is that I sort of started to learn over time, like, who I need by my side when I'm starting a company, because entrepreneurship actually can be lonely. And it's really important to be kind of on the journey, like, with other people that are super like-minded. And these 2 folks absolutely were. So the idea for this company, which is called Satellite Bio, was to take cells from what people had tried to do in the past, which is just infuse them and transplant them, and actually make them little satellite therapeutics. So you leave the sick liver in place, and you would give these cells outside of the sick liver and keep the cells happy, thriving, and surviving in another location, and make it so that patients didn't need transplants. We knew this was going to be expensive. We actually launched the Series Seed right before COVID and then put the financing on pause. And it really favored us in the end, because as some of you will remember, the market was fine actually through the summer of 2020. And you could raise money on Zoom, which meant that we could put together a syndicate, a global syndicate, like from a kitchen counter, which was amazing. So this company raised a pretty large Series, $110 million, recruited a CEO. They're now about 40 people working on their first IND. So this has been really exciting, whole different business model, lots of surprises here as well, and really great like-minded co-founders. So I just want to wrap this all up and give you some of my lessons learned for your own journeys. And that was one way to think about it, is to put this all on a timeline and to show you kind of my evolution as an entrepreneur. So I started out in 2007. That's when the little one was less than a year old. I was raising my babies for a while, and Heprogen as well. And then Glimpse came along. That was the one that I took a sabbatical to run. And then you can see what's happened in the last few years is that I've gotten faster and better, kind of at recognizing when projects that I'm attached to can be startups and really should be advanced to patients through this route. So, you know, in the last few years, we've just gotten better at thinking about how to do this. The common ingredients across all of these are that we had a major publication, a key patent, a trainee who was willing to go into the company and carry all the soft science, all that sort of non-published stuff to really make it work. Really important mentors who are well connected in the sector. And then at some point, every one of these projects had to have an investor who was willing to take a leap, you know, who believed more in us than the risks and just said like, okay, let's give it a go. The uncommon ingredients were the environment. The financial environments have really varied over this time scale. Right now, as you probably heard, it's an epically bad time to start a company. But on the other hand, it's actually a really great time because by the time you need a lot of capital, we're going to be out of it. And my own experience has grown and changed. And, you know, every startup is unique. If you think of them like children, like every single one is a different personality. Things I wish someone told me, it's more fun with others. You know, being with people that you like and trust is really important. You should be flexible because your project will change from what you envision, but also inflexible because investors will push you to do things that they think that they want. And sometimes that's actually not the right thing for the project or not the right thing as you see it. So there's a dance. And then have a thick skin. You just get used to the no's. It's totally different than writing an NIH grant where you like send it off into the ether and it comes back with a score. This is like, it feels very personal. But you know, you just need one yes. So, you just have to have a thick skin and keep going because it's worth it. So with that, I will say that I'm totally inspired by all of you and the day you have ahead. We are doing complementary work at MIT to try and encourage more women in our little ecosystem to start companies because we have noticed and written about how the women at MIT anyway, are underfounding relative to our male peers. And there are all kinds of complicated reasons for that, but we want them off the bench and in the ecosystem through mechanisms like Equalize and some of the activities that we're doing at MIT. So have a great day and thank you so much for including me and cheering you on.